Alright. So listen. So first we will discuss about the syllabus. So the syllabus is the following things. So what do you mean by genes? What is the responsibility of a gene? Can you tell me? What is the function of a gene?
I think it contains hereditary material.
It contains hereditary material. Hereditary material. What do you mean by hereditary material?
They can be passed from.
Yeah, very good. They will be passed from one generation to the next generation, the second generation. Can be, whatever transferring from previous generation to next generation can be stated as a genes. But today we know this one. But imagine we had 100 years previous. We were 100 years ago. So what happening at that time was, biochemists are trying to resolve what is life. So they are trying to understand what is there inside a cell. They came to know inside the cell it has a nucleus and it has a cytoplasm. Then they understood that the cells that has nucleus is always being alive, and dead cells don't have that nucleus. So nucleus, they thought that nucleus is the genetic material, or they thought the nucleus has the life. The cell which has nucleus is alive. Dead cells don't have nucleus. They understood that nucleus plays a vital role. Nucleosome. Then what they did, they started tracking down the biochemistry of this cell. When they broke in the biochemistry, they have found proteins. They found proteins. Then they found some nucleic acids. They found some nucleic acids, and they also found some lipids in the cell membrane. Okay. Now they need to figure out which one is responsible for keeping us alive. Which one is responsible for keeping the cell alive? They still don't know what's happening inside this nucleus. It's still unknown. So they have started doing some experiments initially. So what they did was, there is a scientist called Griffith. So he wanted to prove that DNA is the genetic material. Today it's not a big deal for us, but previously they thought that God interference, nature interference, because of God gift we are getting babies. No one believing that there is something on genetic material that can be passed on. So no one knows this concept. So during this time what happened was, what Griffith did, he observed two types of bacteria. A smooth bacteria, smooth staphylococcus, and another one, rough staphylococcus. Rough staphylococcus. Okay. This is virulent and this is non-virulent. Now you understand what is virulent and what is non-virulent. What does it mean? If we inject this one into a rat, the rat will die. The rat will die. If we inject this into the rat won't die. Simple. Then how this converted to this one? Maybe due to some mutations, the serial killer became a good person. So now there are two types of people. Okay, good bad population and good population. Okay. Then what our Griffith started doing the experiment is, he took this virulent strain, I'm writing here smooth strain. He took virulent strain, that is smooth. Then he heat killed it. He subjected it to a heat. He took a test tube, he took water, in the water he suspended a smooth strain and he heated it. Obviously when we heat something it will die. Right? So the same thing happened. This laptop going to worst. Yeah. So he heat killed the pathogen. Then what happened? Then he reintroduced this suspension, this smooth bacteria to check whether it is died or not. He re-injected into a rat. Then what happened? Rat didn't die. Why rat didn't die? Because this is the time. Experiment was successful. Then, now see the interesting thing. And what he did was, he took these dead colonies, they are dead colonies of this strain, right? He took the dead colonies and also he took the good, rough colonies. These are alive. These are living colonies and this is a dead colony. Pay attention to here, don't worry about the diagram. See, this is a dead material and this is a living material. And the results were shocked when he combined both of them. Okay? Is it possible for a dead person to give his gene to a living person? It's not possible naturally, but that is what's happening. Somehow, the genes, the virulent genes in the dead body transfer to the rough strain. When we introduce this rough strain to the rat, interestingly, rat died. Usually, rough strain won't kill the rat. But how this rough strain became a virulent? That means this indicates that genes can be transferred among the same organisms. This is a living proof that reproduction can happen. That bad genes or good genes. If I have good genes, I can carry bad gene even though the person is dead. There are some mechanisms of uptaking the bad gene into us. This is called as Griffith's experiment. So Griffiths is the first person who stated that genes can be transferable, genes can be hereditary, hereditary can happen. These things can be passed on. Until then, no one believed it, but that's happening in the nature by this experiment. So this is what is called as Griffiths experiment. Is it clear? See, the same thing, whatever I said was pictorially represented here. There is a smooth strain, injected to a rat and it died. Then he took the same smooth strain, mutated to rough strain. So rough strain is a different category. When he injected rough strain, rat was alive. Then he killed the smooth, rat was alive. He took the dead colonies, combined them with the rough colonies, injected it into a rat, and now the dead colony's genes has transferred to the rough colony, making the rough into a virulent strain, and the virulence made the rat to die. So living, yes, recovered. Oh my God! Now this is interesting. See what he said in the last statement. Now this rough became smooth. Rough became a smooth strain.
It mutated or
uptaken, not mutated, it uptaken the dead gene. Not even mutation of it. Oh,
So it integrated it into its own genome.
huh?
Like it integrated the gene from the dead cells into its own genome.
Yeah, and it suppressed the non-virulent gene and its own gene became dominant. Maybe this is a dominant allele and that is a recessive allele. Maybe rough is a recessive allele, but smooth is the dominant allele. So smooth became dominant and that means literally rough became a zombie now. Technically, right? Now it's carrying a dead person's information. Now literally it became a zombie. Now it will start biting other people. Like a very nice strain. Wow, that is really very interesting experiment. So that is Griffith's observation. Okay. Actually, I didn't prepare for this class. I was prepared for something else, and suddenly I'm making. That's why somewhat slow. Anyway, clear? Yes. So this is the first one. So outline the significance of Griffith's transformation experiment. So this is called as the transformation experiment, where the genes has been transferred. Right? So let us see what the ChatGPT said. This is ChatGPT notes. So at the end of the day, you need to know the concept. It has been what books we are using. Right? That is what we do. Don't mistake me that I'm using AI tools. That's not the matter here. Right? See, Griffith says that Griffith transformation experiment conducted in 1928 demonstrated that transforming principle could transfer genetic information between bacteria. He used two strains of Streptococcus pneumonia, a virulent smooth strain and a non-virulent rough strain. This experiment showed that heat-killed smooth strain bacteria could transfer their virulence to a rough strain, transforming it into a virulent form. This finding was a pivotal moment in molecular biology, providing the first evidence that genetic material could be transferred and the DNA might be the hereditary material, because they are still guessing which is the hereditary material. Is it the phosphates, is it the proteins, is it the nucleic acids, or is it lipids? Now this shows that it looks like the DNA might be the favorable candidate. Then what is this Avery's experiment? Who is this Avery? We already have Griffith's proof that DNA is a genetic material. Then what Avery did? Let us see. So definitely these are second generation scientists. Griffith done this experiment in 28. Then it took almost 20 years to recheck this experiment. Then it came in 1944. Griffith. Here we got an Avery. So what Avery tried to say? Avery concluded that DNA is a transforming principle responsible by isolating. This is okay. Could be in the upstream. Let me check the pictures once. What Avery did, Griffiths is okay. Avery declared a McCarthy experiment. Okay. So now they want to pinpoint the exact molecule. So these three people are checking, they are selectively removing. They just removed the lipid and they tried. They removed the RNA, they tried. They removed the DNA, they tried. They removed the lipids and they tried. And they came to know smooth strain extract, no components destroyed, rough strain, mouse died. Polysaccharides destroyed, mouse died. No components destroyed. That means pure one. What is this R strain? Why R is coming in between? Let us see. I need more explanation about Avery's. Let us see. Explain more about Avery experiment. Oh, no, I can't use GPT because it's recorded. Okay. So let us see here. So in Avery's experiment, by isolating DNA, RNA and proteins from pneumonia, Avery and his colleague demonstrated that only DNA could transfer the rough strain. That's it. They are proving that lipids won't do anything, proteins won't do anything, but only DNA killing this one. So they have proved it is the DNA. So molecularly they confirm DNA is the ideal candidate of virulence, not the other biomolecules. Whereas Griffith just said there is a chance of hereditary. He proved hereditary happening. Whereas these people has proved which molecule responsible for hereditary. So this is the second generation scientist. Then who are the third generation scientists? So Avery's conclusion, let us see. Avery said that understanding DNA, DNA is the candidate for the hereditary. That is Avery's conclusion. Avery concluded DNA. Griffith concluded hereditary possible, hereditary happening in the organisms. Then what is this Hershey-Chase experiment? Hershey-Chase experiment conducted. Let us see Hershey-Chase. Alfred Hershey and okay. Let me do something. What they did here? They took a bacteriophage, a virus. Then what is this 35S means? 35S means, any idea? Let me see. Radioactive sulfur. Okay. They have tagged the viral genome with some radioactivity so they can visualize the movement of the genetic material. So they took a virus they tagged the virus DNA with a radioactive light. Maybe we can say they kept a light to the DNA. Then what they did, they purposefully infected this virus to a bacteria. It is called bacteriophage. Why? Because this virus specifically attacks the bacteria. So what happened here? The bacteriophage entered into the virus entered into the bacteria and it injected the DNA. The DNA has a tag. So we can able to watch the tag. Then what he did? Then blended and centrifuged. Then back phage ghost. Most of the radioactivity recorded in the phage ghost plus bacteria. Phage ghost. What is the outcome of this? See, let us see. Use bacteriophages to confirm that DNA is the genetic material. They label DNA of phages with the radioactive phosphorus and the protein code with the They have two things. They have tagged the code with sulfur 35, whereas the genetic material was tagged with P32. Then what they did? After infecting E. coli, they observed that only the radioactive DNA entered the bacterial cells, proving that DNA carries the genetic. Oh, these all people are trying to prove DNA is the genetic material. This is a protein, this is the DNA. So they have seen what this virus will inject into a bacteria. And they came to know that only DNA was injected but not the viral coating. Again proving that DNA is the genetic material. So this is the experiment done by Hershey-Chase experiment. Hershey-Chase experiment. So when they conducted this experiment, they conducted this in 1952. So they have utilized radioactive P32 for DNA tagging, whereas radioactive sulfur 35 for protein coating, and they have utilized bacteriophage for the observation of transfer studies. So let us see the diagram again. Maybe now we can understand. So S35 is the protein code. So protein code was outside the body. Then DNA might be entered inside. We don't know. Then after that, when they separated, when they centrifuged, what happened? In the centrifuge, bacteria was sedimented down, and in the supernatant they have virus, and they have identified radioactivity in the supernatant, indicating that the virus, that is the sulfur 35 present above. That means that is not taken, but here it is nothing. Then what they did? Most of the radioactive ghosts are in supernatant. Then second experiment. They tried, rather than S35, now they tagged with DNA and they did the experiment. Now interestingly, the radioactivity discovered in the sediment, indicating it is the only DNA that is uptaken by the bacteria. That is a very clever experiment. So most of the radioactivity recovered within the bacteria. The viral radioactivity present in the bacteria, because the bacteria has taken this one, viral genome. So indicates that DNA is the genetic material. I think definitely one of these scientists might get a Nobel Prize. They might already got a Nobel Prize. We can see that. So this is all about the third experiment, which is Hershey-Chase experiment. So this experiment emphasizes the importance of DNA in understanding diseases. So they have proved it. DNA carries the genetic information for replication. So next one. So far we have understood that. Can you able to tell me the answer of this question without seeing the answer? What are the key findings of Griffith experiment? What you understood so far about Griffith?
The DNA is a hereditary material.
Now, the DNA is hereditary said by these people. Griffith don't know what is DNA. He is a clueless guy. He is just an experiment with the two bacteria.
Smooth and rough. That's smooth and rough. That was it.
Well, you can try it.
He said like the virulence factor can be transmitted from the smooth strain to the rough strain that is non-virulent and make it virulent.
Yeah, what he felt was there are two types of species, a virulent and non-virulent. Even when we are killing the virulent and we are combining virulent with non-virulent, somehow the virulence of a dead was recovered in the living being, converting the non-virulent to a living, indicating somehow the dead transferred something that is virulent in nature to the non-virulent organism, making the non-virulent to a virulent, indicating there is a chance of transferring the genetic material. Something is transferring between them. You don't know what is that something. Something was hereditating. Something was taken from the previous generation. Okay? Actually, not even previous generation. That is a straightforward thing, parent to children. But here it is, they have cross-checking the genome. I think these are prokaryotes. Prokaryotes don't have a defined nucleus to contain the DNA. So the DNA is freely floating in the cytoplasm. So it is very easy for them to exchange their DNA through the plasma membrane, it seems. So that is why prokaryotes are.
It's a type of reproduction, transformation.
Transformation is a type of reproduction which is only possible among prokaryotes but not in eukaryotes. So we can't expect that thing from humans, like we are touching someone and we are becoming them. Bacteria they don't work. So let us see what the answers state. We know the key findings of Griffith's experiment was the discovery of transforming principle, obviously, where genetic information from heat-killed virulent strain could transform non-virulent rough strain bacteria into a virulent form. Whatever we got, same thing was discussed here. This transformation indicates the existence of transferable genetic material. Right? This is all about the Griffith's experiment. So let us see the next question. Which organism were used in Avery's experiment? So let us see. We need to remember that also. Griffith used Streptococcus pneumonia, right? Streptococcus pneumonia is used by Griffith. Whereas Avery, what Avery did? Can you recall? I forgot. What Avery did here?
He took out parts.
Yeah, Avery just isolated the biology, protein, lipid, and DNA, and he said that only DNA. How he proved it? How he proved that only DNA? He took multiple things. First he tried with protein, then he tried with lipid, then he tried with DNA. Only DNA made killing the one. So okay. So for that what?
Also pneumonia. He used also
He also okay. Avery also used pneumonia. Then what he used? Griffith. Let us see. Oh, both of them use same thing. Streptococcus pneumonia, Streptococcus pneumonia. They use same organism always. That is a simple question, simple answer. So, what is the key findings we wrote it? Which organism were used in Avery's experiment? Same. Pneumonia, rough and smooth strains. Over. Next one. Sixth one. What is sixth question? What is the role of bacteriophages in Hershey-Chase experiment? Can you guess what is the role of bacteriophage in this experiment, Hershey-Chase experiment?
It helps to show that, like, the bacteriophage injects only the DNA material and not the protein code inside the
bacteriophage acted as a carrier of the radiolabeled tag to carry either the gene or the protein code. So they can clearly see which one is right thing. So they used radio tag utilizing the bacteriophage to transfer the gene into the bacteria. So bacteriophage has been used. So let us see. So in the Hershey-Chase experiment, bacteriophages used as a model to demonstrate that DNA is the genetic material. They infected E. coli with phages laden with radioactive isotopes. So showing that DNA, not protein, was passed into the bacterial cells. For that purpose, we need to break down this answer. It's looking too small, right? We need to add these two also. In Hematology, the bacteriophage, the protein code of the bacteriophage was labeled with the sulfur-35, whereas the genome of the bacteriophage is labeled with P32 radiotab. Once they did the experiment, they have concluded that it is the only DNA that is recovered from the bacteria upon doing the centrifugation. That indicates that it is the only DNA that is responsible for hereditary.
They did two experiments so that firstly.
Yeah, they did two separate experiments. First they did protein labeled experiment, and protein was in the supernatant, indicating protein was not transferred in the bacteria, in the sediment.
Then another they marked the
DNA. Now they marked separately they did, not two tags at the same time. Separately they took the P32 phage, then they did the experiment. And second time it recovered in the sediment. So their experiment is straightforward. Whatever recovered in the sediment is transferred. Supernatant they don't know. Because sediment is bacteria. And they recovered that in the bacteria. Correct. So this is all about the sixth question. I think 6 questions we already know. You can directly write in the exam. See? Maybe one question might come if you are writing an MSc one also. Then, what is the central dogma of molecular biology? Actually, I am not taking this topic today. We'll see later that. Any other questions? This I'll take in the afternoon classes. Don't miss the classes. This I'll take afternoon. Only these things. We can work one is easy for you. Now listen, my material is only just guiding material. Don't rely on these notes. You make it better notes. We have seen that there is deficiency in the notes. So this is just for an understanding for you. You prepare notes from checking online or going to library, but you make a better notes because you are master students. I believe you can do it. So I need not to do these things also. But I am helping you to make the learning easy. So make a very good answers and you should impress me with your critical answers, depth in your answers. Understanding topics is easy, it is the basics of it. So can I expect that you will submit notes on time and you will get better in your exams?
Yes, sir.
Very good. So this class is until 10, 20 or 10 more? 10,
10,
10, 20. Oh, then shall I continue? I thought it's finished. Then see, what is the meaning of central dogma of life? Do you have any idea? You don't know. Listen. Central dogma of life means entire planet Earth, whatever living organism we know, it always following this pathway, this direction. That direction is called a central dogma of life, the central concept of life. They are saying that first we have a DNA in the nucleus. This DNA was converting to a RNA. It is converting to RNA. Then this RNA converting to protein. And this protein is making a life possible. Proteins are structures. One of the important things. Then you might ask, what about glucose? How glucose is made from a DNA? If you ask the question, glucose is a made of molecule. We can say like this: we are humans. We cannot make a rice, but we can cook the rice. We are taking other things. We are keeping them together to make another molecule. So DNA utilize the proteins as a tools to prepare all other things. If you don't know, all enzymes are proteins again, but not all proteins are enzymes. You know that statement, right? Then if you think lipids also have a protein actually, lipoprotein. It is the proteins that are keeping lipids in between. So the proteins playing the game of life actually. It is always the proteins that play the game. If you see the DNA, say DNA is a different molecule, how DNA, so at the end of the day, this protein must make another DNA also, that is life. If you see closely, DNA has many proteins tightly packed in between the chromosomes. So always the proteins that are ensuring that these things are passing on. Okay, but the instructions is given by the DNA. So life is very complicated. One way or other. So this direction is called as central dogma of life. DNA, DNA to RNA, RNA to protein, and protein leads to life. This is the central concept. Okay, so conversion of DNA to RNA is called as, any idea? Very good. Transcription. Conversion of RNA to protein is called as? Very good. Translation. Translation. Now you know what is transcription and what is translation. Somehow evolution has favored this directional life but not the reverse. A protein cannot make a RNA and RNA cannot convert to a DNA. It is always this direction only. DNA to RNA, RNA to protein, but not reverse. Why evolution taken this approach? Why an RNA not converting to DNA and DNA making protein directly? Why that is not there? I have understood that, in every copy, when we are making a duplicate, for every 10,000 copies, there will be one error in this pathway. For every 10,000 copies, one error might happen in the RNA. If we are using RNA as a primary template to make a DNA, for every 10 copies, it will make 2 mistakes. Not 10,000. The rate of errors, because RNA is unstable, it's not a rigid structure. So nature obviously favored, because RNA cannot sustain the life. The hereditary, the longevity cannot be possible with RNA because of its weak form. So this is called a central dogma of life. The central dogma of life is conversion of DNA to RNA, RNA to protein, protein to life, we can say. Higher structures first. Okay, that is called a central dogma of life. This concept is called a central dogma. Now we are saying very easily. Initially, scientists are still puzzled with this. They are unable to understand how these things are working. Whether first RNA came or protein came, lipids came, carbohydrates came or DNA came, how they are working each other. What is the step-by-step process? So they have done multiple experiments to conclude central dogma of life. It took them around 25 years to draw the conclusion. So this is not a one-day work actually. For past 200 to 300 years, we are trying to understand. Starting with the oldest guy will be our monk. He is a church pastor. The church pastor made the base for these theories, evolutionary theory and these genetics. Do you know who is that church pastor? Personally, see, we have Newton in 1300s, 1200s. We have Aristotle in 2000 years ago. They are very big scientists, but none of them has discussed about life. But they are understanding mathematics. They understood the universe. They calculated the orbits. They understood the heliocentric theory. But none of them has understood what is life. It is one of the challenging puzzles. And that time, one church pastor, he has a good knowledge of statistics. So what he was doing is, he is having pea plants in his garden. When he was doing his gardening, he is observing something. There are yellow peas and black peas. He is thinking, why these black peas are coming? Then he took a yellow pea plant, black pea plant, and he grafted them, and he saw the pollen, next generation. Then he is thinking 50-50 ratio. He has Punnett square. He made a Punnett square and he is checking 50-50 ratio. And he started understanding the ratios of hereditary. He don't know what's happening in the plant. He don't know the molecules, nothing. But he's just seeing the plant babies, the offsprings. Okay. And you know what is his name? Whom I'm saying? John Needham. John Needham is different, not John Needham. And even I forgot his name. The pea plant experiment, who did it? He is a famous name. Google his name. Pea plant experiment, genetics. He is a pastor actually. He don't have any biology background, but he made the new subject, so-called hereditary.
The one who said that recessive and that.
Recessive allele dominant. Mendel is pea plant experiment. John Mendel and after saying this, I wish it's okay. See, Mendel's experiments made this field. This is very interesting field actually, biology. How it's just a natural puzzles. They are trying to solve a rubric cube. Still the rubric cube is still complicated. We try to understand one phase of colors, but still there are many jigsaw puzzle on the other phases. Alright, we'll see in the afternoon. Thank you, Marcel.
Thank you.